All right, everyone, we are live. A very good afternoon and warm welcome to Shri Chaitanya, everybody. This is your NEET 10 Game Changer series, wherein we are going to discuss 25 different concepts and 25 different questions every day in physics, chemistry, botany, and zoology. This is your botany class. I'm your botany faculty, Beena Yadav. And I welcome all of you to this super amazing session. We are going to cover 25 different concepts again, as we are doing on daily basis. So in today's session also, there will be 25 different concepts, and these 25 different concepts are picked up from the latest collection, latest data of PYQs, the concepts from which every year repeatedly question has been asked. I have picked up all those concepts and framed questions on those particular concepts, and we are going to solve questions on those particular concepts. All right, everybody. So first thing first, do hit the like button, do share it with all your friends, because it is going to be a rapid fire round kind of session, because today is 29th of April. We are left with just 4 to 5 days, and we just need to quickly revise all those concepts. We need to quickly see, like, the question might be asked from this particular concept in such a way, okay, because we have different type of questions that we see. Either it is rise the column type of question, either it is statement based question or one-liner question, or we can also expect fill in the blanks type of questions. All right, everybody. Yes, hello Vinod, hello Shreya, hello Yasu. Do hit the like button and share it all your friends. Come on, share it with all your friends and quickly join the session. So this is your NEET 10, a game changer series, which claims that you're going to get 650 plus marks in NEET 2024 based upon this series, because we have specifically picked 25 questions per session, and these are the most important 25 different concepts of entire botany portion. All right. So this is your timetable of the day. You already had 2 classes. This is your third class that I'm going to take up, 3 p.m. to 4 p.m. That is for botany, and after which you're going to get class of chemistry, 4:30 to 5:30. Pawan sir will be taking care of your chemistry portion. All right. So let's get started, kids. Let's get started with the very first question of today's session, which is in front of you. This question is from the genetics part. All right. Genetics being the most important unit of your NEET examination, I would say the weightage is nearly around 18% in genetics. We do have principle of inheritance and molecular basis. Both the chapters are very, very important. And if I segregate between these 2 chapters, then molecular basis is something which is having high importance. Reason being, in 2023 8 questions were asked, in 2022 7 questions were asked. So that way it makes this particular unit very important and very high weightage type of unit. Okay, I can see the answers of this very first question. Anish, Jyoti, Amrita, come on everybody, keep on trying. Vinod, hello, answer, come on everybody, answer. The question is from Turner syndrome. An example of monosomy is, and the answer will be option number A, which is Turner syndrome. See, if I speak about chromosomal disorders, my dear kids, if I speak about chromosomal disorders, so be it your Down syndrome, number 1, be it your Klinefelter syndrome, number 2, be it your Turner syndrome, number 3, these are very, very, very important. In 2022, question was from Klinefelter syndrome. Previous year, question was from Down syndrome. So all these syndromes are really important. You can expect one question from these 3 syndromes for sure. Either it is on the trisomy of 21 chromosome, which is your Down syndrome. Either it can be from Klinefelter syndrome, or it can be from Turner syndrome. But one question will definitely will be there from this particular portion. All right. Yes, Amrita, Jyoti, Anish, can we have plus for the people who have answered it correctly? Can we have plus for everybody? Answer is option number 1, which is Turner syndrome. That represents X0 condition. So basically we have 1 less chromosome in the people who are having Turner syndrome. All right. So plus 4, and moving on to the next question, kids. Next question is on sickle cell anemia. Disorder arises due to, what is the reason for sickle cell anemia? Sickle cell anemia, the disease in which your RBC's shape is different, that is sickle shape. From normal shape of RBC, the shape of RBCs will be sickle shape in such a way that they are not able to. Very good, Anish. Very good. Very good, Amrita. Very good, Anish. Perfect, perfect. Rest of the kids, come on, answer quickly, quickly. Yes, sickle cell anemia, Jyoti, very good, is basically because of a point mutation. Point mutation. So specifically, if I speak about, we have change in just one nucleotide. If we have change in just one nucleotide, so your amino acid has been changed. So from glutamic acid, that should be the normal condition. The condition is now we have valine. Okay. So due to the change of this one base pair, or we can say change of this one amino acid, the entire shape of RBCs has been changed. Okay, so it arises due to substitution in a single base of DNA, just one nucleotide change, and that will result in different amino acid, and that will result in entire different type of protein, and that will result in entire different shape of RBCs. All right, so answer will be option number C, that is substitution. There is no duplication, there is no deletion, because despite one nucleotide, we have different nucleotide, okay? There is no duplication at all. So answer will be your option number B, that is substitution in a single base of DNA. All right, is this clear? Can we have +4? Come on kids, +4 in this question, and moving on to the next question. +4 in the previous one, and let's move to the next question. The association of two subunits of the ribosomes in translation is governed by concentration of what? Okay, in ribosome, we do have two subunits. We have small subunit, we have small subunit, and we have large subunit. All right. So basically, we need to unite our small subunit and the large subunit. Yes, great, great, great. And for association of these two subunits, we do need some ions. Very good, very good. This is Mg2 plus, kids, Mg2 plus, calcium 2 plus, I mean Ca2 plus, which is calcium. It is H plus, and it is ATP. Which ion is required? Amrita, Jyoti, Girija, Vinod, come on, answer has to be. Great answers, it is magnesium. Magnesium ion is required for association of two subunits, which is large and small subunit, all right. So answer has to be option number 1, which is magnesium. +4, and moving on to the next one. Come on. Next one, grasshopper is an example of X0 type of sex determination in which the male have. See, Hanking sir, at first he discovered sex chromosome in insects, specifically grasshopper, okay. So that is example of X0 type, which means we have X0 condition, X0 and XX. Okay, this is typical case of male heterogamity. Your male would be having one chromosome less than the female. Okay, female is XX, male is X0, which means 50% of the insects will get X chromosome. Rest 50% of the male will not get any chromosome at all. Okay, yes, Niga. Answer, Jyoti, Anish. Great, great, great. So basically, it is an example of X0 type. Males have one X chromosome. X0 condition is there. Alright, so male would be having one X chromosome and female will be having two X chromosome. That is why your female is having similar, two same chromosomes. Okay, so we cannot say this is the case of female heterogamity. We would say it is a case of male heterogamity. Alright, so answer has to be option number 1, which is 1X chromosome. Great, everybody. Let's move to the next question then. Next one on your screen. Next, kids, a character having the inheritance pattern which depicts same phenotypic and genotypic ratio in F2 generation. You have to define a character in which you're going to get same phenotypic and genotypic ratios. Come on. Good afternoon, Yasu. Good afternoon. Next question. Come on, Anish. All right. Rest of the kids, Come on, come on. We're going to get same phenotypic and genotypic ratios. Same ratio for both phenotype as well as genotype. Okay. Hello, Shriyash Yadav. You don't know, Anish, not a problem. C option, okay. Rest of the kids. Okay, let me explain the question because nobody is correct in this question. Okay, you people are saying it is option number C. So my dear kids, if I speak about Pisum sativum, that is pea plant, in case of pea plant, the F2 ratio, the genotypic ratio comes out to be 1:2:1. All right. However, the phenotypic ratio would be your 3:1. Okay, in case of all the monohybrid crosses wherein we are taking into account just one character, be it your stem height, be it your seed color, be it your seed shape, any character, but that has to be monohybrid. In that case, the ratio will be, genotypic will be 1:2:1, phenotypic will always be 3:1 ratio. Okay, but the question is asking you about the phenotypic and genotypic, both the ratios should be similar. That is a typical case of what? Typical case of incomplete dominance, my dear kids. If I speak about incomplete dominance, which is found in the case of snapdragon flower color. Okay, let me give you the complete cross of flower color of snapdragon. So basically in the P1 and P2, parent 1 is having red colored flower, parent 2 is having white color flower. This is red, this is white. All right. In F1 progeny, capital R will come from parent 1 side, small r will come from parent 2 side. So we are having capital R and small r. Now, due to the presence of incomplete dominance, this is having pink color flower. Pink colored flower. Okay, if it would have been dominance, in that case the color has to be red color, but your snapdragon shows incomplete dominance, which means your dominant color is not completely dominant and your recessive is also not completely suppressed. So you're gonna get a color which is intermediary color. That is why in case of F1 progeny we have pink color flower. Now question is asking about F2 generation, right? So if I'm crossing between F1 and F1, that will be my capital R small r into capital R small r. If I'll draw the checkerboard, which is this one, I'm having capital R small r, capital R small r. In the first box, capital R small, small r, small r. All right, so carefully analyze. This is my pink color flower, this is my white color flower, again pink color, this is my red color. Okay, then let's find out the genotypic ratio. If I speak about genotypic ratio of this one, so we can have three different combinations: capital R, capital R small and small r, small r. Just count the number, everybody. Just count the number. 1 capital R, 2 capital R small r, these two, and 1 is small r, which is this one. Right? Let's find out the phenotypic ratio. So we can have again three different combinations. One is red flower, second is pink flower, and third is white flower. Okay, so red flower is just 1, pink are 2, 1 and 2, and white is again 1, which is this one. Can you people see the genotypic and phenotypic ratios are similar? Yes or no? Yes or no? Yes, kids, tell me. Can we see both the ratios are similar? Yes, we can definitely see. So question was asking you about The character having the inheritance pattern which depicts same phenotypic and genotypic ratio, that will be flower color in snapdragon, and the ratio will be 1:2:1. Correct? Alright, so +4 or -1? +4 or -1? Come on, next question then. Come on kids, next question on your screen. A person with O blood group have his mother and father with A and B blood group. Okay, so mother is having A blood group, okay, and father is having B blood group, okay, and person is with O blood group. All right, what would be the genotype of both mother and father? What should be the genotype, kids? Anish, okay, Jyoti, great, Amrita, perfect. Rest of the kids, come on. Yes. Yes, Anish, Arya, perfect answer. Yes, so see, it is the A blood group. Carefully understand, if the genotype could be either IA or it could be IA small I. All right. If the genotype is this one, which means the contributing gamete from the mother can be just one, which is IA. Okay, but my O blood group is result of small I, which means one I is coming from the mother, second is coming from the father. Okay, so obviously this genotype cannot be of your mother, it could be IA small I. Similarly, if I speak about father, we can have two different combinations, IB, which is homozygous, and we have IB small I, which is heterozygous. Alright, if I speak about IB small I, again, the progeny is having O blood group, which is result of homozygous condition, which means from mother this one I has come, from father this second I has come. So obviously in the genotype of father also we should have the heterozygous condition, which is IB and small I. So the conclusion is mother and father, they both should have heterozygous condition. Okay, so mother and father both are heterozygous for A and B blood group respectively, which means this is IA small I, this has to be IB small I. Is this correct? And as Anish has answered it correctly, if it is not adopted, then of course this should be the genotype. Alright kids, perfect. Let's move to the next question then. Next question with your +4 and -1. Next question, mycorrhiza promotes plant growth by. Mycorrhiza promotes plant growth by. What is mycorrhiza first of all? It is symbiotic association of fungi with the roots of higher plants. That is my mycorrhiza. Okay, so myco is simply your fungi and rhiza is basically your roots of higher plant, right? So it is symbiotic association between fungi and the roots of higher plant. So basically they both get benefited out of each other. The plant has got capacity to photosynthesize. It will give glucose molecule, which is food for the fungi, as well as shelter to the fungi. Fungi is having hyphae. It can spread its hyphae to a longer distance and absorb water and mineral. So fungi will give water and mineral by spreading the hyphae, and your plant will give food and shelter to the fungi. That's why this is mutualism, this is symbiotic association, right? Okay, I'm getting the answers. Great, great. Yes, Sue? I'm getting the answers. Come on, come on. Promotes plant growth. How come mycorrhiza promotes plant growth? Protecting the plant from infection, or it can helping the plant in utilizing atmospheric nitrogen, serving as plant growth regulator. It is absorbing inorganic ions from the soil because it has got hyphae which can spread to a longer distance, and if it will spread to the longer distance, they will be able to absorb water and minerals, and by absorbing water and minerals, it is capable to give it to plants. Okay, so correct answer will be option number 1. Good afternoon, Neer. Correct answer will be option number 1, which is A option. Right, everybody? All right. So correct answer is 1 plus 4. Next, move to the next question, kids. This is match the column type of question. This is little time taking question. Try your level best to solve the question in very less time also. Okay. So we have column 1 and column 2. In column 1, we have F0 component of ATP synthase. So in ATP synthase, we have F0 and F1. There are two components. F0 is the one which is spanning the entire membrane, and F1 is the one which has got clubbed with ATPase synthase. ATPase synthase is basically the enzyme which is responsible to make ATP. Okay. Yes, great. I'm getting the answers. Answer is option number 1. See, so F0 component, NADH, FADH2, oxygen or integral membrane protein. It is integral membrane protein. Your F1 component is basically the peripheral one. Okay, this is protruding outside and coupled with your ATP synthase enzyme also, which is going to make ATP, okay? Then final hydrogen accepted during electron transport chain will be oxygen. Oxygen will receive electron as well as hydrogen and will form metabolic water. So terminal electron acceptor we can say, or final hydrogen acceptor we can say, would be your oxygen only. Source of proton during electron transport chain is your NADH and FADH2. Your 1 NADH2 will give rise to 3 ATPs, whereas your 1 FADH2 will give rise to 2 ATPs to us. Okay. Okay. Yes. So correct answer has to be option number 1. Option number 1. Okay. Let me explain it one more time if you have confusion. So this is basically your F0 and F1 component. Okay, this is F0 component, this is F1 component. Alright, this F0 is spanning into the membrane like this, integral membrane protein, that's why we are saying, and this is the peripheral one. Okay, this F1 is clubbed with your ATPase enzyme like this. Right, what this ATPase enzyme will do, this will pick up ADP plus PI and will make ATP. Okay, so the process is whenever 2H plus has been passed from this F0 F1 particle, there will be structural conformation into F0 F1, and whenever there is structural conformation into this F0 F1 particle, your ATPase will get activated. Whenever your ATPase get activated, it will make ATP molecule. Okay. Okay, I hope it is clear now. So let's move to the next question. Kids, do hit the like button also. Okay, do hit the like and share it with all your friends. Come on everybody, next question on your screen. Come on, next one. Accumulation of proton in chloroplast during photosynthesis. Okay, during photosynthesis, there is accumulation of proton in chloroplast occurs at which step? Very beautiful question. I'm getting the answers already. Yes, Neer. Yes, Jyoti. Great. Rest of the kids, come on, answer. Yes, Sue. Perfect. Rest of the kids, come on, answer. Accumulation of proton in chloroplast during photosynthesis. See, if I speak about ATP generation, so ATP formation is basically due to the presence of proton gradient, or we can say due to accumulation of proton motive force. Okay, proton gradient is nothing but the difference of the H plus concentration. Where is the difference? In lumen as well as in stroma. In case of lumen of thylakoid, we have more number of H plus than the stroma. So basically, this increased number of H plus into the lumen will create this proton motive force. We need to create a balance. In order to create the balance, we will take help of F0 F1 particles. When your H plus will move from F0 F1 particle from lumen to the stroma, then your ATP synthase enzyme will get activated and finally ATP will be formed. So that's how your process is happening. Okay, so basically there has to be proton gradient, and if I speak about proton gradient inside the lumen, the concentration of H plus is more than the stroma. In case of stroma we have less H plus concentration. In case of lumen of thylakoid we have more H plus concentration, thereby making a gradient of H plus, and when we are going to break this gradient in the process we will make ATP. Okay, yes, so correct answer has to be option number B, which is inside the lumen. Correct, everybody? Yes, let's hit +4 in this question and moving on to the next question then. Next question, in a flower, if the megaspore mother cell forms megaspore without undergoing meiosis, and if one of the megaspore develops into an embryo sac, its nuclei would be. This is purely an hypothetical question. Okay, nothing that's sort of happening actually. Okay, it is just to check your knowledge about the ploidy level. So let's say if in a case we have megaspore mother cell which is forming megaspore, so actually it forms megaspore when there is meiosis, or we can say reductional division happening. Okay, now question is saying there is no meiosis. If meiosis would have happened, in that case it is my diploid and it will make haploid megaspores. Yes, let's say we do not have any meiosis here. Then what will happen? The number of chromosome will not be reduced to half. The number of chromosome will remain same, which means if it is 2n here, it will definitely be 2n only. Okay, it will not be reduced to half. So your question is saying, if let's say there is no meiosis and if one of the megaspore develop directly into embryo sac, okay, there is no meiosis involved, then of course the ploidy will be your diploid. It will be your diploid only. Okay, because meiosis will lead to a reduction of the number of chromosomes just into half. So 2n will convert itself into n. Here if meiosis is not there, then of course it will remain 2n. Okay, alright kids, so answer is option number B, which is diploid. +4, let's hit a +4 and moving on to the next question. Next one kids, the phenomena wherein the ovary develops into a fruit without fertilization, that is called as what? The ovary has developed into fruit and there is no fertilization involved. What do you call? Do we call it parthenocarpy? Do we call it apomixis? Do we call it asexual reproduction or sexual reproduction? These two options are garbage, I would say. You might get confused into apomixis and parthenocarpy, although I think there should not be any confusion. Okay, Moksh, I got your answer. Rest of the kids? Neer, okay. Amrita, Anish, okay. Yes. Alright kids, it is your parthenocarpy. The definition of parthenocarpy simply is the phenomena wherein the ovary develops into a fruit without fertilization. So your ovary is 2n, okay. So ideally what happens, your ovary has got meiosis and we have a formation of eggs into the ovary, which is haploid, and the male gamete is also haploid. So when there is mixing, or we can say fusion of this male and female gamete, both are haploid, then we get diploid. Okay, so if let's say in a condition your ovary, which is diploid already, has gone for fruit formation without any fertilization, then that is called as parthenocarpy. Okay, so Yesu, yes. Nazneen, yes, answer will be option number 1, which is parthenocarpy. Alright everybody, I guess maximum of you got +4 in this question. +4, can I see +4s everybody? Can I see +4s? Okay, great. Next question on your screen. Next question, which molecule is found in both sorghum and rice? So in case of sorghum as well as rice, you're going to get one molecule, okay, regards to photosynthesis. Your sorghum is C4 plant, C4 plant. What is that one molecule that is common for both C3 and C4 plant? You'll get your answer. Great answer, Jyoti. Great answer, Anish. Avinash, think about it. Yes, 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 perfect answer. What is that one thing that is common for both C3 and C4 plant? My dear kids, if I speak about C3 plants, okay, in case of C3 plants, obviously my Calvin cycle, or we can say C3 cycle is happening. That is the major source of formation of glucose molecule. Yes, okay. But if I speak about C4 plants, in case of C4 also, there is this one step called as CO2 fixation happening into mesophyll, which is outermost cell. For this CO2 fixation, the responsible enzyme is peptase, all right. But after which there is formation of glucose also, which is getting fixed into bundle sheath cells. In case of bundle sheath cells, we do have presence of rubisco, all right, which means your Calvin cycle is happening. Your Calvin cycle will be present into C3 as well as C4 plants, which means the common product of C3 and C4 would be there in both the cases. That is my phosphoglyceric acid, because this is specific for C4 plant, right? This is exclusive for C4 plant. This is also exclusive for C4 plant. My phosphoglyceric acid, which is the first product of Calvin cycle, will be there in both the plants, C3 as well as C4 plant. So answer will be option number A, which is 3-phosphoglyceric acid. Hello, Arjun Reddy. Yes, Shriyash Yadav. Is this clear, everybody? Is this clear? Moksh got 0 in this question. Rest of the kids, Anish got +4, Amrita got +4, Neer +4, +4. This is clear to everybody? Yes. All right. Moving on to the next question then. This question is based upon sex determination. Very important topic. If I speak about genetics, one question will definitely come from this particular topic. Very important topic, okay? Yes, yes, come on, come on. So we have organisms and the type of sex determination. Grasshopper, insect, we're gonna get X0, male heterogamety or female heterogamety or haplodiploid system. In case of fruit fly, we have male or female heterogamety. In case of humans, we have male or female. In case of honeybee, what will be the case? Getting the answers already. Bhargav, madam, how many questions we can expect from ecology? Bhargav, see, since in 2024 environmental issues has been removed, 1 or 2 questions used to come from that chapter also. So we can say in total from entire ecology we might expect 8 to 9 questions in total. Okay, 8 to 9. Your pyramid part is really important. Your type of population is very important. Population density is equals to natality, mortality, immigration, emigration. That is very important. Okay, adaptation part is important. Migration, suspension, estivation, diapause. That is very important. Okay, so these parts are very important from your ecology portion. Okay, yes, yes. Come on, come on, come on. This question solve, everybody. So first of all, what is a male heterogamety? If your male has got 2 different type of chromosomes, we call it male heterogamety. In case of human being, we have XXXY condition. This is my female, this is my male. Male has 2 different type of chromosome. I would call that male heterogamety. Okay, but if let's say ZZZW, my female has 2 different type of chromosome, my male has got 2 similar chromosome. This is what we call as female heterogamety, found in the case of birds. Okay, so female in case of birds and male will be in the case of humans. In honeybee, we're gonna get haplodiploid system, wherein your male is always haploid and female is always diploid. Okay, in insect we have X0 type of system. Everybody is correct, I can see. Yes, Amrita, Anish, Neer, Jyoti, Shivu Kumar, Mizna, great, great, great. Yes, it would be yes to option number C. +4, can we hit +4 for this question, everybody? Yeah, we can do so. +4 for this one, and moving on to the next question. Yes, this part from ecology, one of the students was asking about ecology. This is really, really important. This is basically interspecific interactions, very important portion from ecology, my dear kid. Okay, your examiner can ask question like this one, which is match the column type of question. We can expect one-liner question also. We can expect a question from examples of all these interactions also. Okay, so this is really important. Anish, okay, Neer, okay, Amrita, Jyoti, come on, rest of the kids. Rest of the kids, come on, answer. Shivu Kumar, very good. Very good. Your competition, the two species which are involved for competition will both get negatively impacted. Negative effect would be there, okay? Amensalism, one is negatively impacted, one has no effect at all. Predation, we have one predator, one prey. One is positive, second is negative. I can see positive and 0 has been written. This is incorrect match, okay? Then parasitism, again one is plus, one is minus, which is accurate, right? So correct answer will be option number C. Question has asked about incorrect match. That would be option number C. All right, everybody? Yes, it is option number C, which is incorrect. Clear? Great, +4s, and let's move to the next question then. Next one. In angiosperms, the parenchymatous mass of tissue enclosed within the integument and forms the body of the ovule is called as. There is this parenchymatous mass enclosed within the integument, okay? So this is my ovule, right? And this is my outer integument, this is my inner integument. This is my outer integument, this is my inner integument, right? Then we have this central portion wherein there is presence of mass of the cells. This mass of the cells is there. This one is called as nucellus. Okay? Okay, kids? Yes? All right, everybody, this is nucellus, right? Answer is option number D. Perfect. +4, and moving on to the next question. Okay, kids, if the session is, you are finding it fruitful, then please do hit a like button also and subscribe to our very beautiful channel, because such classes will be there for you people till your NEET examination. Okay, we will be running these classes up to 4th of May so that you can have, a quick revision of your all the subjects, be it botany, zoology, physics or chemistry. All right, so please do hit the like button and share it with all your friends, and subscribe our channel and press the bell icon also, okay? You will get notified for all the classes which are there. Come on, next question on that note. Next question, kids. Next question on your screen. Very nice. Transgenic variety of rice, that is golden rice, has been developed to overcome the deficiency of which vitamin? So that is basically made to overcome the deficiency of vitamin A. Okay, so answer will be option number D, which is vitamin A. Right? Yes, answer is option number D, vitamin A. I can say many correct answers. Rudra is there, Moksha is there, Amrita is there, Neer, Shivu, Jyoti, Anish. Great. It is option number D, which is vitamin A. Perfect. Perfect. Shreyash Yadav. Great. Next question. In which of the following organisms gametes produced are isogametes? Gametes which are produced are isogamete. See, we can have 3 different conditions. We can have isogamy, we can have anisogamy, and we can have oogamy. These are the 3 different conditions. Getting the answers perfect. Please, people, Write down your answers. Let me explain what is isogamy. The very first type is isogamy, okay. Then we have number 2, which is anisogamy. Anisogamy. Then we have number 3, which is ogamy. Okay, we can have 3 different types, right? So starting from isogamy, the gametes which are involved, iso means similar, okay. Gamy means a fusion. So the gametes which are there, both the gametes are, they are similar, okay, similar. Now similar in terms of everything, similar in terms of size, similar in terms of shape, everything would be there. If the gametes are motile, both will be motile, or if they are non-motile, both will be non-motile, okay. So the similarity will be in such a way, if both the gametes involved, if they have motility factor, both will be motile. If they do not have, both will be non-motile. Now moving on to the second one, which is aniso. Aniso means different, okay, and gamy is again fusion. So the gametes which are involved are different. Both the gametes are different. Difference could be in terms of your size, in terms of the shape, in terms of motility. One is motile, second is non-motile, like that, okay. And third is your oogamy. In case of oogamy, we're gonna get a specific type wherein my male gamete will always be small in size and it will be motile. It is small in size and motile. And my female gamete will always be, large in size. It will always be large in size and it will be static in the position. It will not move at all. Okay. So in case of oogamy, your male gamete is always small and motile and female is always large and static. Now you would say both the gametes are different in this case also. Then how come it is different from anisogamy? The thing is, the two gametes which are involved, male and female, the difference in on the basis of size, shape will be marginal difference. There is not such a huge difference. Okay, you can take a reference of human being. In case of human being, the male gametes are released at once, are trillions of male gamete, very tiny, and they are motile. And the female gamete is very large comparatively and static. Okay, so that difference of the size, which is humongous, that will be in the case of oogamy. But if there is a marginal difference in the basis of size, that is my anisogamy. And if the size is almost similar, that will be my isogamy. Okay, is this clear, everybody? Is this clear, everybody? Yes? Is this clear, everybody? Great. So my question is saying, in which of the following organisms gametes produced are isogamete? Now isogamete, which means both the gametes could be motile or both the gametes could be non-motile also. Okay, and this example has been given into my NCERT, which is Cladophora. Cladophora has got isogametes. Okay, yes, Rudra, clear? Answer will be option number A, which is Cladophora. I hope it is clear to all the kids out there. Yes? Alright. So +4 in this question and moving on to the next question then. Next question on your screen. Come on. Next question on your screen. Next question, kids. For the formation of 40 embryo sacs, how many total number of meiotic division are required? If I want to make embryo sac, how many meiosis are required? Neer, okay. Amrita, okay. 1/4, okay. Anish. Rest kids, come on. If I speak about formation of embryo sac, let me take you to the megaspore mother cell. Let's go back to megaspore mother cell. In case of megaspore mother cell, your meiosis will happen, okay, and it will form, of course, from one megaspore mother cell, which is my diploid, we're gonna get 4 megaspore that will be haploid, okay. We have 1, we have 2, we have 3, and we have 4. So these are the 4 megaspores which have produced megaspore, correct? And they all are haploid, correct? But the irony is, out of these 4 megaspores which have produced, 3 will degenerate and only 1 will be functional. Out of these 4, your 3 will degenerate and just 1 is functional. The one which is functional, that will make my embryo sac. That will make my embryo sac. So I can clearly see from one megaspore mother cell, my one functional megaspore will be produced, and from one functional megaspore, one embryo sac will be formed. So per meiosis, I'm going to get 1 embryo sac. Question is saying we have 40 embryo sac. Then tell me how many total meiosis would have happened. So it would be your 40 only. Okay, because per meiosis I can see just 1 embryo sac has been formed. Because in total 4 megaspore has formed. We could have 4 embryo sac, but out of 4, 3 would degenerate, only 1 will be functional. The one which is functional, that will make your embryo sac. The 3 will be degenerated. Is this clear, everybody? Yes. So answer will be option number A, that is 40, 40. Clear? Nice question. Thank you very much. Moksh, is this clear to you? Anish, clear? Amrita, clear? Yesu, Shriyash Yadavji, is this clear? Yeshu, I don't know Tamil. Please, I would really appreciate if you can write your question in English. If you have any request, any doubt, I would really be happy to, take care of your doubt if you can write in English. Okay, because my Tamil is a little on the poor side. Okay, perfect. Moving on to the next question then. Next question, come on. If a DNA molecule consists of 25% of guanine, then amount of thymine present is. This question is based upon Chargaff rule, okay, based upon the Chargaff rule. Come on, try to solve the question. The question is saying the content of guanine is 25%, okay. Now find out the content of thymine. How much thymine would be there? Jyoti has answered, great. Amrita has answered, great. Anish has answered, perfect. Rest of the kids, come on. Come on, come on. Neer has answered, very nice, very nice. Perfect answers, everybody, perfect. So according to Chargaff sir, basically your A plus T plus G plus C, your purine plus pyrimidines should be 100%, alright. Now, this guanine is 25%, this is my 25%, and we know guanine always binds with cytosine, which means if it is 25%, obviously it will also be 25%. So guanine is 25, then cytosine is also 25. Then we need to find out adenine plus thymine plus 25, 25, that will be 50 is equals to 100. A plus T will be your 50. Okay. Now the amount of adenine will always be equal to amount of thymine because A will bind to T with the help of 2 hydrogen bonds. So we can say this 2T or 2A, one and same thing, that is my 50, my T is 50 by 2, that is 25%. So the content of adenine is 25, content of thymine is 25, and of course guanine is 25, cytosine is also 25%. The answer will be option number C, which is 25%. Alright, it is 25%. Clear, everybody? Is this clear to all my kids out there? Yeps. Clear? So +4 for this question, and let's move to the next question from cytology. Very important question of cytology. We're gonna get one question for sure from this particular portion also. We have column 1 and column 2. In column 1 we have metacentric, we have submetacentric, acrocentric and telocentric. Based upon the position of centromere, we have divided our chromosome into 5 different categories, which are metacentric, submetacentric, acrocentric and telocentric. Okay? Please do answer. Please do answer. Let me explain the question also. Okay? So based upon the position of centromere, my dear kids, based upon the position of centromere, which is basically your primary constriction, we can have 4 different type of chromosomes. Number 1 is your metacentric. Number 1 is metacentric. Number 2 is your submetacentric. Submetacentric. Number 3 is your acrocentric. Acrocentric. And number 4 is your telocentric. It is telocentric. Correct? We have 4 different type of chromosomes. Let's see what is the difference among these 4 different types. So the first one, which is metacentric. This is my P arm, this is my Q arm, and we can see the centromere has divided the chromosome into 2 equal halves, which means if your centromere is at center, if it is at center, we call it metacentric. In such a way, my one arm is equal to the second one, which means my P arm will always be equal to Q arm, because centromere is exactly at the center position. Okay. Then second, we have submetacentric. Now, my centromere has shifted on the upper side. So, this is my centromere, this is my one arm, and this is my second arm. So, centromere has shifted away from the center. We call that submetacentric. We can see my P is not equal to Q. My one arm is longer than the Q. Okay. What about acrocentric? My centromere has shifted more on the one extreme end. So this will be something like this. We can see my P is greater than Q. Okay. Then we have the telocentric. If my centromere is at tip, in such a way, there is no P, just Q is there. Okay. If my centromere is at tip position, we call it telocentric. So we can have metacentric, wherein my centromere is at central position, wherein my one arm is equal to the second arm, P is always equal to Q. Second is submetacentric, wherein my centromere has shifted its position on the upper side from the center. So P is not equal to Q certainly, and P is higher than the Q. Okay. Third is my acrocentric, wherein centromere is little bit on the upper side. Okay. And in telo, the centromere is at tip position. Okay. Yes. Yes, everybody. Yes. So now come back to the question. Metacentric, center of the chromosome. Okay. Telocentric, terminal centromere. All right. Close to the end, acrocentric, slightly away from the center is submetacentric. So it will be option number C. A is 3, B is 1, C is 4, and D is your 2. Yes? Is this clear now? Not a problem, Munni. At your own convenience. Is this clear now to all the kids out there? Yes, it is clear. All right, moving on to the next question then. Next question, kids. The synthesis of sugar during biosynthetic phase of photosynthesis occurs in sugar. Biosynthetic phase is your dark reaction, okay? It is your dark reaction wherein your CO2 is actually fixed into glucose molecule. Clear? Your CO2 is fixed into glucose molecule. Clear? This will be happening into your stroma because all the enzymes are there into the stroma. So all the enzymatic activities will be happening inside your stroma. So your CO2 will be fixed into glucose into stroma only. Correct answer will be option number C. +4 for this question, and let's move to the next one. Pretty easy question, and you will surely get questions based upon their scientists and the contribution from the genetics part. Okay. The mapping of genes on chromosomes using the recombination frequency between gene pair as distance between them was performed by. Who did the mapping? Yes, great answer. Anish, Jyoti, Amrita, Yesu, come on, Mizna, come on everybody. It was the effort of Alfred Stuyvesant sir, who happened to be student of Morgan. Okay, you know, we say the type of company where you live in, basically you get the character of that company. Okay, yes, so his teacher was working on Drosophila melanogaster, his teacher was working on linkage and all the sort of genetics problems. So that drew his attention also, and he just thought that just by finding out the distance between the genes, we can actually map them. Okay, we can do a mapping system for them. Yes, it is Alfred Stuyvesant. So correct answer is option number B. Please put a +4, and moving on to the next question. Probably the last question, I guess. Yes, this is your last question. Respiratory roots growing vertically upwards in the plant of swampy areas are known as. This question is from morphology. Very important question. The type of roots, which we also call them respiratory roots, they are little different from the normal roots because the normal roots are positively geotropic and negatively phototropic. These are just opposite. These are positively phototropic and negatively geotropic. Okay, and there is presence of nematothodes, which are pore-like structure responsible for respiration. Yes. Great! It is called as nematophores or respiratory roots. Okay. From this portion, please do remember about number 1, what are respiratory roots, called as nematothodes also. Secondly, the plants which are able to have these respiratory roots, which are your Rhizophora, Avicennia, Sonertia. Number 3, they are found in swampy areas, and number 4, they can show vivipary also. So these 4 points are really important from this particular portion. Okay, so your respiratory roots are nematophores. Done, everybody? Yes, Janaki. Yes, Rani. Anish, Jyoti, Amrita, Neet. With this, your 25 questions, we have completed our 25 questions, and I hope you liked the session. If you liked the session, then please give a thumbs up to our efforts. Hit the like button, share it with all your friends, and subscribe our beautiful channel. We'll see you in the next session tomorrow, same time, 3 p.m. to 4 p.m. Daily basis we are having classes. Tomorrow we will again meet 3 p.m. to 4 p.m. Alright kids, bye-bye, and be happy, be healthy, take care of yourself. We'll see you in tomorrow's session. Until then, take care. Bye everybody. Thank you so much for having me. Bye.